Imagine you pull an all-nighter. Maybe you are studying, maybe you are working late, maybe you are scrolling way too long, maybe you just cannot sleep. The next morning, you already know you are going to feel tired. But here is the question. Is tiredness the only thing happening in your body? Or does one sleepless night already change the biological signals that control hunger, fullness, and metabolism? That is exactly what a laboratory study published in Obesity tried to investigate. The researchers asked a simple but powerful question. Can one night of total sleep deprivation change hormones related to appetite and body weight regulation? And the answer was yes. At least in this study, one night without sleep shifted several hormones in a direction that may make hunger regulation harder. Not enough to say one bad night makes you gain weight, but enough to say sleep loss is not just mental fatigue. It can also be a hormonal event. When people talk about weight gain or fat loss, they usually talk about calories, diet, exercise, and willpower. But the body is not just a calculator. The body has signals. It has hormones that tell the brain, I'm hungry. I'm full. We have enough stored energy. Go find food. And when sleep is missing, those signals may change. This matters because many people who sleep badly also notice something very real the next day. They feel hungrier, they snack more, they crave more calorie-dense foods, and they often have less energy to move. So the question is not, does one sleepless night magically create fat? The better question is, does one sleepless night shift the body toward a state where eating more becomes more likely? This study looked at that question by measuring 3 hormones: leptin, ghrelin, and adiponectin. First, leptin. Leptin is often described as a satiety hormone. It is produced mainly by fat tissue and sends signals to the brain about energy availability. A simple way to say it is, leptin helps tell the brain, we have enough energy stored. So if leptin goes down, the satiety signal may become weaker. Second, ghrelin. Ghrelin is often described as a hunger hormone. It is produced mainly by the stomach and helps signal hunger to the brain. A simple way to say it is, ghrelin tells the brain, go find food. So if ghrelin goes up, the hunger signal may become stronger. Third, adiponectin. This one is more complicated. Adiponectin is produced by fat cells and is often associated with metabolic health, insulin sensitivity, and anti-inflammatory effects. But in this article, the authors explain that animal studies suggest adiponectin may also act in ways related to food intake and fat storage. So adiponectin is not simply a good or bad hormone here, it is more complex. And that is why the researchers measured all 3. They wanted to know whether one night without sleep changes the hormonal environment connected to appetite and metabolism. The study included 44 young adults. Their average age was 24.9 years. There were 20 women and 24 men. The researchers also included people from 2 weight groups. 25 participants had normal weight and 19 participants had obesity. The obesity group was defined as having a BMI of at least 30 kilograms per square meter plus elevated waist circumference. The normal weight group had a BMI below 25 kilograms per square meter and normal waist circumference. The participants were generally healthy. They did not have acute or chronic physical or psychiatric diseases. They were not shift workers, and they had not travelled across time zones in the month before the study. That last part matters because shift work and jet lag can strongly affect sleep and circadian rhythm. The researchers wanted to isolate the effect of one night of sleep loss as much as possible. This was a randomized crossover laboratory study. That sounds technical, but it is actually easy to understand. Each participant came to the sleep laboratory for 2 experimental nights. On one night, they were allowed to sleep. On the other night, they had to stay awake all night. Because every participant did both conditions, each person was compared to themselves. That is a strength of the study. Instead of asking, are people who sleep less different from people who sleep more? The researchers asked, what happens inside the same person after sleep versus after no sleep? The two sessions were separated by about 1 week. Before the real experimental night, participants also had an adaptation night in the laboratory. That helps reduce the first night effect, where people sleep differently just because they are in a new place. The experiment was controlled. Participants arrived at the sleep laboratory and were supervised. At 7 p.m, they received a standardized dinner. That matters because the researchers did not want one person eating a huge meal and another eating something totally different before hormone measurements. In the sleep condition, participants were allowed to sleep from 11 p.m. to 7 a.m, so they had an 8-hour sleep opportunity. Their sleep was measured using polysomnography, which is a laboratory method used to record sleep. In the sleep deprivation condition, participants stayed awake the entire night. They were continuously supervised and could do quiet activities like watching movies, reading books, or playing board games with the experimenter under normal indoor lighting. Then, in both conditions, after the night was over, blood was collected at 7:30 a.m. The blood sample was taken after an overnight fast. Then the researchers measured fasting levels of leptin, ghrelin, and adiponectin. So the comparison was very direct. Same person, same lab, standardized dinner, 1 night with sleep, 1 night without sleep, blood test the next morning. The first major finding was that leptin decreased after total sleep deprivation. Remember, leptin is linked to satiety signaling across the whole group. After the sleepless night, leptin was 17.3 nanograms per milliliter. After the normal sleep night, leptin was 18.6 nanograms per milliliter. That is about a 7% decrease. The result was statistically significant, with p equals 0.037. So the simple story is, after 1 night without sleep, the satiety-related hormone leptin was lower, but we need to be careful. This does not mean every person became extremely hungry. It does not mean 1 bad night causes weight gain. It means that 1 biological signal related to fullness was reduced the next morning. The second major finding was even more intuitive. Ghrelin increased after total sleep deprivation. Remember, ghrelin is linked to hunger. Across the whole group, after the sleepless night, ghrelin was 839.4 picograms per milliliter. After the normal sleep night, ghrelin was 741.4 picograms per milliliter. That is an increase of about 98 picograms per milliliter. The result was statistically significant, with p equals 0.003, so after 1 night without sleep, the hormone associated with hunger was higher. This is probably the most powerful storytelling point, because a lot of people have experienced this. You sleep badly, and the next day, you want more food. Not always because you are physically starving, sometimes because your brain and body feel off. This study gives a possible biological explanation. After sleep loss, the hunger signal may become stronger. The third finding was that adiponectin increased after sleep deprivation. Across the whole group, after the sleepless night, adiponectin was 7.5 micrograms per milliliter. After the normal sleep night, it was 6.8 micrograms per milliliter. That is about a 10% increase. The result was statistically significant, with p equals 0.003. This part needs nuance. Adiponectin is often described as beneficial for metabolic health, but the authors explain that some animal research suggests adiponectin may also act like a starvation-related signal, and may be involved in increasing food intake or promoting lipid storage in fat cells. So let's put the 3 findings together. After 1 night of total sleep deprivation, leptin went down. That means the satiety-related signal decreased. Ghrelin went up. That means the hunger-related signal increased. Adiponectin went up. That suggests a metabolic response that is more complicated, but potentially relevant to appetite and fat storage mechanisms. The overall pattern is 1 sleepless night shifted the endocrine balance away from satiety and toward hunger. That does not mean a person automatically overeats, but it may help explain why poor sleep often makes food choices harder the next day. The researchers also looked at whether the response was different between men and women. For leptin, the decrease was statistically significant in women. In women, after sleep deprivation, leptin was 25.8 nanograms per milliliter. After normal sleep, leptin was 28.1 nanograms per milliliter. That result had p equals 0.030. In men, leptin was 10.1 nanograms per milliliter after sleep deprivation and 10.6 nanograms per milliliter after normal sleep. That was not statistically significant, with p equals 0.458, so it looked like the leptin decrease was mainly visible in women. But here is the important nuance. The direct comparison between men and women did not reach statistical significance. For adiponectin, the increase was also statistically significant among women. In women, adiponectin was 9.4 micrograms per milliliter after sleep deprivation and 8.4 micrograms per milliliter after sleep. That result had p equals 0.025. In men, the values were 5.9 micrograms per milliliter after sleep deprivation and 5.6 micrograms per milliliter after sleep. This was close, but not statistically significant, with p equals 0.056. Again, interesting pattern, but not something to overstate. The researchers also compared participants with normal weight and participants with obesity. The clearest subgroup result was for Ghrelin. Among participants with obesity, Ghrelin was 750.4 picograms per milliliter after sleep deprivation and 629.9 picograms per milliliter after normal sleep. That was statistically significant, with p equals 0.007. Among normal weight participants, Ghrelin was 913.0 picograms per milliliter after sleep deprivation and 833.5 picograms per milliliter after sleep. That was not statistically significant, with p equals 0.095. So the increase in Ghrelin appeared especially clear in the participants with obesity. But again, we need caution. The obesity subgroup included only 19 people, so the correct interpretation is the study suggests that people with obesity may show a more noticeable Ghrelin response after sleep loss, but this needs to be tested in larger studies. This matters because it raises a bigger research question. Could sleep loss affect appetite-regulating hormones differently depending on body weight status? This study suggests that possibility, but it does not settle the question. Now let's be very clear about the limits. First, the study does not prove that one sleepless night causes weight gain. The researchers measured hormones, they did not measure how much food people ate afterward, so we cannot say they had more Ghrelin, therefore they ate more. That is plausible, but this specific study did not directly test food intake. Second, blood was measured only once in the morning at 7:30 a.m., so we do not know whether these hormonal changes lasted all day. They might have faded later, or other effects might have appeared later. This study gives a morning snapshot. Third, the study had 44 participants. That is useful for a controlled laboratory study, but still small, especially when you split the sample into men versus women, and normal weight versus obesity. Fourth, the study tested total sleep deprivation. That means a full night without sleep, but many people in real life do not stay awake all night. They sleep 5 or 6 hours, so we still need more research on partial sleep restriction, sleep fragmentation, and real-world sleep patterns. So what should we take from this paper? Not that one all-nighter makes you gain fat, not that sleep replaces nutrition, not that hormones override calories. The real takeaway is more subtle. Sleep loss can change the hormonal environment that regulates appetite. After just one sleepless night, this study found lower leptin, higher Ghrelin, and higher adiponectin. In simple terms, less satiety signaling, more hunger signaling, and a broader metabolic shift. That means poor sleep may make weight management harder, not because it breaks energy balance, but because it may make appetite control more difficult. So the question is not, does sleep magically burn fat? The better question is, does sleep loss make it easier to overeat? And this study suggests, biologically, it might. So the next time you hear someone say weight control is only about willpower, remember this. Willpower happens inside a body, and that body is not the same after a good night of sleep as it is after a sleepless night. After one night without sleep, the body may send weaker fullness signals and stronger hunger signals. That does not remove personal choice, but it changes the conditions under which those choices are made. Sleep is not a magic solution, but it is not a detail either. It is part of the biology of hunger.